Have you continuous evolution? New variations of the system such as CRISPR-Cas12 and Cas13 have been discovered. These variations offer different capabilities and advantages, expanding the possibilities of genome editing. CRISPR-Cas12, for instance, targets both DNA and RNA, giving researchers more flexibility. Meanwhile, Cas13 is unique in that it targets RNA exclusively, which can be advantageous for tackling diseases related to RNA. With this knowledge, we can now explore the practical applications of CRISPR in plant genetics. A CRISPR has made significant contributions to enhancing crop resilience and nutritional improvements. Let's consider how this powerful tool has been used to bolster the hardiness of our food sources. CRISPR technology has been applied to engineer crops that can withstand the brutal onslaught of drought, pests, and diseases. It's like giving plants their own suit of armor designed at the molecular level. Now imagine a world where staple crops like rice and wheat are not just filling but nutritionally fortified. That's another frontier where CRISPR is making waves. By tweaking the genetic makeup of these plants, we're able to enhance their nutritional content, a process known as biofortification. But what about the ethical and regulatory considerations? Well, CRISPR-based precision breeding is more targeted than traditional GMOs, reducing the risk of unintended genetic changes. This makes it a more accepted method of genetic modification. But the power of genetic engineering doesn't stop with plants. Let's see how it's used in animal genetics. From disease models to gene therapy, CRISPR has revolutionized animal genetics. Imagine a world where genetic diseases are a thing of the past. This isn't science fiction, it's a reality that's being shaped right now. By using CRISPR, scientists can precisely edit the genes of animals, potentially eradicating inherited diseases and improving animal health. But it's not just animals that are benefiting from CRISPR. Microbial engineering is another exciting frontier. By editing the genes of bacteria and other microorganisms, we can harness their abilities for good, creating new medicines and industrial products. And let's not forget about our planet's endangered species. Genetic engineering holds promise for conservation efforts, with the potential to bolster populations of critically endangered species. As we venture further into the realm of genetic engineering, we must also consider the ethical, legal, and social implications. How do we balance the benefits with the potential risks? These are questions we'll explore in the next scene. The power of genetic engineering comes with a weighty responsibility. As we wield the power to rewrite the genetic code of life, we're treading on uncharted ethical territory. Genetically modifying organisms isn't simply a scientific question. It's one that touches on our core values and principles. How far should we go in modifying life? Could we inadvertently create organisms with unforeseen consequences? These are not easy questions, but they're ones we must grapple with as we continue to push the boundaries of this technology. The ethical debates surrounding genetic engineering are multifaceted. Some argue that it's our duty to use these tools to improve human health and secure our food supply. Others express concerns about playing God and the potential for unforeseen ecological impacts. The truth likely lies somewhere in between, but it's a conversation we all need to be a part of. On the legal front, the regulatory landscape for genetically engineered organisms is equally complex. Different countries have different regulations, some stricter than others. In the United States, for instance, the FDA, USDA, and EPA all have a say in the regulation of genetically engineered plants and animals. These agencies are tasked with ensuring that these organisms are safe for us and for our environment. Yet regulation can be a double-edged sword. While it's undoubtedly necessary to ensure safety, overly stringent regulations can stifle innovation and impede the development of potentially life-saving technologies. Striking the right balance is a challenging but crucial task. Lastly, there's the matter of public perception. Despite the promise of genetically modified foods, many people remain skeptical. Misconceptions and fear often cloud the conversation, making it difficult for the public to make informed decisions about these technologies. It's therefore crucial that scientists and educators communicate the benefits and risks of genetic engineering in a clear and balanced manner. Now that we've navigated the complex landscape of genetic engineering, let's look at some real-world examples. Scene script. Real-world examples provide a tangible understanding of the potential and limitations of genetic engineering. Let's delve into some successful CRISPR projects. In agriculture, scientists have engineered crops like rice and wheat to be resistant to pests, diseases, and harsh environmental conditions, resulting in higher yield and healthier food. In medicine, CRISPR has been used to treat genetic diseases. A notable example is the application of CRISPR to cure Leber's congenital amaurosis, a rare genetic disorder causing blindness. But it hasn't been all smooth sailing. Failures and setbacks have taught us valuable lessons. For instance, the initial excitement over the potential of CRISPR gene drives to suppress mosquito populations was tempered by the discovery of resistance development in these insects. These examples underline the importance of robust scientific scrutiny and the need for caution as we navigate the complex world of genetic engineering. As we look to the future, we can only imagine what possibilities lie ahead in the field of genetic engineering. The future of genetic engineering holds exciting prospects. Innovations in plant and animal genetics are paving the way for a new era of synthetic biology, where organisms are designed to perform specific tasks such as bioremediation or producing pharmaceutical compounds. We're also engineering plants and animals to withstand extreme weather patterns, contributing to food security in the face of climate change. And as we delve deeper into the genetic code, we're discovering ways to restore lost biodiversity and bolster ecosystem resilience. But the possibilities don't stop in the fields and forests. We're on the cusp of enhancing human abilities through genetic engineering. Imagine a future where we can optimize our cognitive and physical capabilities, where we can extend our lifespan and improve the quality of life in our later years. We're not just talking about preventing hereditary diseases and optimizing health, we're talking about revolutionizing healthcare itself. However, as we stand on the brink of these advancements, we must also grapple with their ethical, legal, and societal implications. The power to alter the genetic code comes with great responsibility. Balancing the benefits of genetic enhancements against potential societal inequalities and ethical dilemmas is no small task. We're in uncharted territory, and developing global regulatory frameworks that ensure safety, ethical compliance, and access equity is paramount. Moreover, we must foster an informed public dialogue on the future of genetic engineering. It's crucial to address fears and misconceptions, and equally important to ensure that the advancements we're making are for the benefit of humanity as a whole. This isn't just about what we can do, it's about what we should do. As we continue to push the boundaries of what's possible, genetic engineering promises to revolutionize our world in ways we can only begin to imagine.